This section introduces the whole chapter of genome editing tools as a single idea seen from several angles. Every tool here is one machine at different resolutions. Each one cuts the double helix at a chosen address, then lets the cell's own repair machinery, either error-prone rejoining or template copying, write the edit. CRISPR, meganucleases, ZFNs, TALENs, and CRISPR. Cas differ in one respect only: how that address is specified. The chapter's arc is a single decoupling. Targeting drifts out of the cutting protein and into a swappable module until CRISPR relocates it onto RNA base pairing instead of re-engineered contacts between protein and DNA. That is why CRISPR closes the sequence. It turns the question of where to cut into typing a sequence rather than folding a protein. Read the survey as where to cut into typing a sequence rather than as 5 separate inventions. This section explains the single move underneath all of gene editing. No editor writes DNA directly. The move is subtractive. Cut both strands at one chosen site, then surrender the break to the cell's own repair machinery. One path is NHEJ, which relegates sloppily and leaves gene-disrupting indels. The other is HDR, which copies a template you supply. So the whole chapter is one search, a cheaper reprogrammable way to point the cut. Meganucleases, zinc finger nucleases, and TALENs each re-engineer protein to recognize each new sequence, costly per target. CRISPR wins by moving recognition off protein onto a guide RNA that base pairs its target. RNA that base pairs its target. RNA that base pairs its target. Only comes a type string, where the older tool, it differs only in how it steers the break. This section explains how the CRISPR-Cas system is classified and why the axis of that classification matters more than its timeline. 6 types, numbered 1 through 6, fall out of that single split, and this one's axis is the interference machine. Class 1 distributes DNA cutting across a complex of many Cas proteins. Class 2 collapses that same job into one large effector, Cas9. 6 types, numbered 1 through 6, fall out of that single split. That split is the surprise the chapter's history sets up. Bacterial adaptive immunity was found by accident in 1987 as odd repeats sitting near an E. coli phosphatase gene. It was read as phage memory only by 2005 and reprogrammed in 2012. But the toolkit the whole book builds rests entirely on Class 2's collapse. A single programmable protein is what turned the immune system into an instrument. Where Class 1 needs a committee of Cas proteins, Class 2 needs one. Architecture is the real taxon. This section explains how a whole family of defense systems collapses into a usable taxonomy along a single axis. A defense system remembers its enemies by filing captured viral DNA between palindromic repeats. Every CRISPR type shares this memory, which splits the whole zoo, all 6, 2, 5, and 6, into a usable taxonomy is a single bit. Does the interference machine that finds and cuts the target run as a multi-protein complex, which is Class 1, covering types 1, 3, and 4, or as one self-contained protein, which is Class 2, covering types 2, 5, and 6? That bit is the spine of the chapter. It is also why Cas9 became the tool. Engineering wants one deliverable protein, not a committee. History supplies the sequence of noticing. Classification supplies the axis, effector cardinality, along which everything downstream is indexed. This section explains why a programmable editing platform turns guide design into a computational problem. Programmability carries a hidden cost. Once the Cas protein is fixed, hardware and only the 20 nucleotide guide varies. Editing specificity is decided before any molecule exists. It collapses to a search problem over the genome. A guide that pairs with its intended site will also pair with every near-duplicate sequence a tolerant nuclease accepts, so the chapter's whole toolkit is in. Silico scoring, predict on-target cleavage efficiency, enumerate off-target sites, rank candidate guides. That is what this chapter is for in the book's arc. After Chapter 2 catalogs the Cas machinery, here, the wet lab act of editing becomes a computation. The risk now sits in the design of the guide, and the enzyme is no longer where it lives. This section explains why precision in CRISPR editing has to be computed rather than trusted to the enzyme. A nuclease you can aim at any 20 letters also fires at every near copy of them. The very shortness that makes CRISPR programmable is what makes it promiscuous. So the enzyme does not hand you specificity. You compute it back. Between mechanism, meaning what Cas is, and application, meaning what you edit, sits an unavoidable middle layer, the guide design tool that scores which 20mer cuts hard on, target, and rarely off target. The primitive is this. Whenever recognition is short and mismatch tolerant, precision migrates out of the wetware and into the search algorithm. The chapters before this one forge the scalpel. This one concedes that the scalpel is blunt until software sharpens it. This section explains the 4 delivery methods for CRISPR-Cas reagents and why the whole problem is one of logistics rather than chemistry. The scissors are solved. Getting them into the nucleus is not, and that inversion is the chapter's whole point. Cas9 and the single guide RNA are fixed, but the payload can be encoded at 3 depths along its own production line. You can deliver it as DNA, which the cell must transcribe, then translate, then traffic the protein back to the nucleus. You can deliver it as messenger RNA, which the cell only has to translate. Or you can deliver the ready-made protein RNA complex directly. Each step you move downstream buys transients, briefer action, fewer off-targets, no integration risk. It pays for that with harder delivery, because the molecule you are now moving is bigger or charged. Orthogonally, a vehicle carries whichever form you chose, and the vehicle can be physical, viral, or non-viral. Every named method is 1 point on a single surface, persistence against safety, against how easily the cargo crosses 2 membranes. Delivery is a logistics problem. The chemistry is already solved. This section explains why, in genome editing, the remaining hard problem is delivery rather than the cut itself. The cut is solved. The courier is not. Once Cas9 became a fixed reusable protein steered by a swappable guide, the hard problem stopped being what to edit and became how to edit, and became how to get the scissors inside, across the membrane, into the nucleus, inside. That is the whole chapter. The bottleneck is delivery, and editing is no longer the hard part. The step that does the work is that cargo form and vehicle are one coupled choice, a single decision rather than 2. The cargo can be DNA, messenger RNA, or a pre-assembled ribonucleoprotein. Each form dictates which carrier can hold it and how long it lingers. Persistence buys editing, efficiency at the price of off-target cutting. Every method downstream is 1 point on that single triangle: reach, safety, and duration. This section explains how the CRISPR-Cas9 system gets re-engineered. CRISPR-Cas9 arrives as a found object, a bacterial immune nuclease that cuts wherever a short guide RNA and an adjacent PAM motif jointly permit. The chapter's move is to treat that windfall as a machine with 2 knobs, nature left coupled and crude. The first knob is where it can cut, the targeting range, gated by the PAM. The second is whether it cuts wrongly, the fidelity. measured as the off-target rate. Re-engineering is the systematic decoupling of the two. Protein engineering relaxes the PAM constraint. Guide RNA chemistry and mined orthologs, Cas12 and Cas-X, sharpen specificity until each knob becomes independently tunable. Every section is one axis of the same optimization. The borrowed enzyme is refactored into a programmable instrument. This section explains what sets the specificity of a programmable nuclease and the knobs that tighten it. A programmable nuclease is a search and cut machine. Its flaw is that the search tolerates near misses. Cas9 cleaves sites that merely resemble the target, and this chapter is the space of knobs that tighten that tolerance. The primary lever is this. Specificity is set by how rare the recognition key is. Cas9 must find a short flanking motif, the PAM, beside its target. The motif NGG recurs every 8 base pairs, but a longer motif like NNNNN, GATT, only every 128 base pairs, so lengthening the key leaves the genome G8 base pairs, so lengthening the key leaves the genome geometrically fewer sites to miscut. Every re-engineering move, orthologs, guide RNA edits, dead Cas9, fusions is one trade. You constrain recognition and rarefy off-targets at the cost of reach. This section explains why every CRISPR tool beyond genome editing comes from a single realization about Cas9. Cas9 does two separable things. It finds a 20 nucleotide address, a guide RNA base paired to the locus, licensed by a neighboring PAM, and it cuts there. The chapter turns entirely on noticing that only the first is the reusable primitive. Kill the nuclease domains, giving dead Cas9, written dCas9, and what remains is a programmable protein that homes to any sequence you name. It is now a bare docking platform. Onto that platform, you fuse the cargo of your choice, a repressor which gives CRISPR-I, an activator which gives CRISPR-A, an epigenetic writer, a base editing deaminase, a fluorophore, or a lineage barcode. The double-strand break was never the essence. Addressing was, every beyond editing tool is one move. Keep the RNA-guided targeting, swap the payload. This section explains why the chapter treats CRISPR as an addressing system rather than a cutting tool. Cas9 does two separable things. It finds a locus and it cuts. The whole argument of the chapter is that the finding is the primitive and the cutting is merely the first payload bolted onto it. Kill the nuclease and you get Cas9, a programmable RNA address delivery vehicle to any point in the genome. Then you fuse whatever cargo you like, a transcriptional activator, a deaminase, a reverse transcriptase, a fluorophore, a barcode. Every beyond the chapter lists is the same move, wearing different cargo. CRISPR interference and CRISPR activation, epigenome writing, base and prime editing, imaging, lineage tracing. One primitive, many payloads. Here is the inversion. The chapter turns on. CRISPR is an addressing system, and cutting was only one instance of what you can do once you can address a locus. The earlier picture of it as essentially a pair of scissors was a single use mistaken for the whole capability. This section explains why RNA editing with CRISPR and Cas13 targets the working copy of a gene rather than its permanent source. Edit the working copy and leave the master untouched. The genome is a persistent archive. Messenger RNA is a continuously retranscribed working copy, so any change to the transcript is inherently transient and dose-tunable, decaying as the cell rereads the unchanged source. That asymmetry is the chapter's thesis. Targeting RNA buys reversibility and tunability for free, and it sidesteps the autoimmunity of cutting your own genome. Cas13, a Type6 system, is the instrument. It is a key RNA-guided ribonuclease whose 2 HEPN domains cleave a matching transcript, redirecting CRISPR's programmable addressing from DNA to RNA. The boundary that reveals the structure is this. A mutation that is lethal or promptly corrected at the genome, useless to touch there, becomes a legitimate target the moment you aim at its transcript. This part explains how a single substitution turns the CRISPR system into an RNA editor. Keep the CRISPR architecture, one programmable guide, one class II effector, and move its target off DNA and onto RNA. That single substitution is the whole chapter. Cas13 is what falls out. It is type 6, it carries a heparin domain, and it cleaves single-stranded RNA under a guide, CRISPR RNA. It reads a protospacer flanking site, a PFS, where Cas9 reads a PAM. The structural payoff is reversibility. DNA edits are permanent scars on the master copy, but RNA turns over, so editing the transcript is dosage tunable and leaves the genome untouched. This is the safer knob that the book's DNA chapters cannot offer. OneTel predicts every application. Once activated, Cas13 keeps chewing nearby RNA indiscriminately. This is collateral cleavage, a nuisance for editing and the entire engine behind Sherlock Diagnostics. This section explains the single move behind rewriting the genetic code with CRISPR. The moment you break Cas9's blade, what remains is the most valuable half, a programmable address. The guide RNA never cut anything. It only located the site by base pairing. Disable that nuclease, giving you dead, Cas9, or the nickase, nCas9, and the same targeting machinery becomes a docking pointer to any chosen letter of the genome. Now bolt on a different cargo, a deaminase that chemically transmutes one base into another in place, and you rewrite rather than sever. This is the chapter's whole move, editing without the double-strand break, escaping the cell's stochastic repair lottery, NHEJ, or HDR, entirely. Cut and pray becomes point and rewrite. Everything downstream, base editing and prime editing, is one substitution on the cargo slot. This section explains why scaling genome editing is a different problem from making a single edit. Editing one locus is surgery. Rewriting a genome is surge. The chapter's single move is this: scale comes not from a bigger cut, but from escaping the cut entirely. A cell tolerates only 4 to 7 simultaneous double-strand breaks before apoptosis fires. The DSB budget is the binding constraint, and every technique here roots around it. MAGE feeds oligos to the lagging strand at the replication fork, mutating without breaking. CAGE conjugates pre-recoded genomes together, merging edits made elsewhere. Evolve-R nicks once and lets an error-prone polymerase diversify locally. These are 3 answers to one question. How do you recode hundreds of codons when you cannot cut hundreds of times? This section explains the single primitive behind every plant, CRISPR application in the chapter. One programmable protein, steered by a swappable 20-nucleotide RNA, is the whole of it. Every application the chapter catalogs, model species, staple crops, horticulture, legumes, trees, is that same primitive pointed at a different genome. Trees is that same primitive pointed at a different genome. The survey reads as a list but threads one thesis. Because Cas9 can be multiplexed and delivered DNA-free, a targeted deletion becomes indistinguishable from a spontaneous mutation. That indistinguishability is the step that makes the whole argument go through. It lifts the edited crop out of the GMO frame entirely, and the USDA declines to regulate a GMO frame entirely, and the USDA declines to regulate a genome carrying only precise deletions. The surprise is regulatory rather than molecular. Programmability buys precision, and it also buys a change of legal category. This section explains one primitive, one address, many payloads. That is the whole chapter. CRISPR's guide RNA fixes where. Whatever you fuse to, Cas fixes what happens there. Cut, activate, repress, rewrite a base, prime, edit, and indel. The chapter is the book's plant instance of that primitive. One programmable recruitment mechanism sweeps model species, crops, horticulture, legumes, trees, crops, horticulture, legumes, trees, changing only the target list, not the machine. Chapter 9 puts it this way. CRISPR brings DNA, RNA, and protein close together in a programmable and predictable pattern. That closeness is the invariant. Each species just supplies new loci. Multiplexing extends the address to a whole set at once. The reach scales with target diversity, not method complexity, which is why a single toolkit can rewrite an entire kingdom's traits. This session explains why CRISPR came to dominate genome editing and why the very trait that won the field is also its central weakness. CRISPR won because it is monomeric. A single Cas9 guide complex does the targeting, where the older tools, ZFNs and talins, need two proteins binding in tandem. That same trait is its defining flaw. Here is the trade-off at the heart of it. Two independent binding events multiply specificity combinatorially. One binding event does not. So a single guide tolerates up to 6 mismatches and cuts where it should not. This is off-targeting. The whole architecture of the chapter follows from this one trade-off. Specificity limits, delivery, ethics, and every future prospect sit downstream of it. The simplicity that made CRISPR dominant is exactly what makes it imprecise. The frontier is buying precision back. Engineered Cas variants, paired nicases, and in silico guide design all aim at this, without surrendering the monomeric ease that won the field in the first place. This section closes the toolkit by turning it on itself. Every virtue that made CRISPR win is the same mechanism that makes it leak. A single short guide, RNA finding its target by base pairing, is cheap, fast, and programmable. And precisely because recognition is one event over about 20 nucleotides, it tolerates mismatches and cuts where it should not. Off-targeting is the tax on programmability. CFNs and talons pay less of that tax because they are dimeric, a structural advantage rather than a cleverer design. Two independent recognition events multiply into a stricter address. CRISPR's monomeric simplicity buys reach at the cost of precision. Read every downstream fix as one move. Paired nicases, FOLCID, Cas9, high-fidelity variants, base and prime editing. Each of them reintroduces a second constraint to recover the specificity that simplicity spent.